everyone, I am Snigdha Bharadwaj, Assistant Professor, ITS College of Pharmacy. Welcome to the presentation. Today we will discuss about various compartment models in biopharmaceutics and pharmacokinetics. So, without wasting much time, let us get started. Basically, pharmacokinetic models is used to study various pharmacokinetic parameters. A basic type of model used in pharmacokinetics is known as compartment model. Compartmental models are categorized by the number of compartments needed to describe the drug's behavior in the body. So, basically in this compartment modeling, we generally assumes body as a compartment and then analyze its pharmacokinetic behaviors. There are one compartment, two compartment and multi-compartment models. So, based on the compartment numbers, we termed it as one compartment, two compartment and multi-compartment models. So, the compartments do not represent a specific tissue or fluid, but may represent a group of similar tissues or fluid. If we talk about blood, so blood is uniform all over the body and we can consider it as a single compartment. These models can be used to predict the time course of drug concentration in the body. So, why we have established why one have established these models to identify the drug concentration in the body at particular time. That means, we can correlate it with the AUC, Cmax and Tmax. These are pharmacokinetic parameters. The one compartment model linear assumes that the drug in question is evenly distributed throughout the body into a single compartment. When we describe one compartment model, we usually assume that the body is a uniform compartment and the drug is evenly distributed all over the body. So, this principle follows in one compartment model. So, from this figure you can identify what I am trying to say. This model is only appropriate for drugs which rapidly and readily distribute between the plasma and other body tissues. So, before administration and after administration, we assume that the drug is evenly distributed all over the body and is uniform. So, we can say that uniform distribution of those drugs which are rapidly and readily distributed between the plasma and other body tissues. Yes, of course, you must remember all these concept of basic pharmacokinetics, that for the first blood is reaching to plasma, systemic circulation, and then it is distributing through to body tissues or peripheral tissues. So, we also observe that first it is coming to plasma and then distributed to various body tissues. So, when we consider body as single compartment or a single blood as single compartment, so it comes under one compartment model. Drug which exhibit a slow equilibration with peripheral tissues are best described with a two compartment. For example, the drug profile is not very rapid, it is a kind of slow. So, we generally we take it in two compartment modeling profile. So, before administration the drug is not entered yet, and after administering, immediately after administration. So, first it will go to plasma and then it is slowly distributed among peripheral tissues, right. So, this the study of two different compartment, that one blood is a central compartment and how it is going to peripheral tissues will be like different kind of compartments we consider, and the study of this two different compartments comes under two compartment modeling. So, you see that the concentration and time profile curve, right. So, the solid line shows the serum concentration time graph for a drug that follows one compartment, solid line, and the two dashed line represent, this line represent the one compartment modeling, that means the drug reaching to systemic circulation at which point of time, it is uniform, constant, linear, whereas the dashed line represent the serum concentration time plot for a drug that follows two compartment modeling pharmacokinetics after intravenous bolus is given. So, few deviation you see here, the plasma reaching to plasma and then distributing to the different kind of body tissues, two different components. So, there is variation or deviation from the linear line is observed in this particular graph. Drug movement within the body is a complex process. So, the drug movement through various processes like absorption, distribution, metabolism and excretion is a complex process. The major objective is therefore to develop a generalized and simple approach to describe, analyze and interpret the data obtained during in vivo drug deposition studies. So, there is a need to develop a particular model type or a system to identify how the drug is depositing to in the body tissues or it is getting distributed among the body tissues, right. The two major approaches in the quantitative studies of various kinetics processes of drug deposition in the body are model approach and model independent approach. We can say model dependent approach and model independent approach, also called as non-compartmental analysis. In non-compartmental analysis, we do not consider body as a compartment or different compartments, and we generally observe without considering it as compartment. So, we will discuss it later. Pharmacokinetic model approach. First, we will focus on the model approaches. So, pharmacokinetic model approaches, a model is a hypothesis that employs mathematical terms to concisely describe quantitative relationship. So, it is a basically hypothetical thing, a model which is a mathematical formula based calculations, and these concisely describe how the quantitative relationship, how the drug is affecting the different processes or it is reaching to the target site. Pharmacokinetic model, basically in this pharmacokinetic model, it provides concise means of expressing mathematically or quantitatively time of course of drug throughout the body and compute meaningful pharmacokinetic parameters, because without any theoretical aspects we cannot prove the experimental data. I hope you understand that if we have some sort of mathematical arithmetic calculation, then we can express and we can correlate it with the experimental data we have observed, and then only we can prove our point that yes, what we have said and what we have observed, that both the data is matching and we can expect that drug performance is quietly relevant to the mathematical data. So, methods of analysis of pharmacokinetic data. We are saying that drug first go through absorption phase, then distribution, metabolism and excretion, but these models based calculation prove it right. Yes, drug is first absorbed through that particular AUC, Cmax, Tmax concentration, plasma concentration, time profile curve. So, all these are basically theoretical aspects we are taken into consideration, and then we are performing the biological experiments and then correlating the results from both the models, from both the aspects, and then compiling the results and interpreting the results and then giving the conclusions, right, whether it is achieving or not. So, methods of analysis for pharmacokinetic data. When we need to observe or assess pharmacokinetic parameters, so we need to set up some sort of mathematical calculations for that, and based on that particular calculations we can perform our experiments accordingly. So, we have different kind of analysis methods like model approach, model independent approach. In model dependent approaches are like it is further subdivided into three approaches: compartment modeling, physiological models and distributed parameter models. So, in compartment models, mammillary method or models is there and catenary model, and in physiological perfusion limited model, diffusion limited models and distributed parameter model. So, these are basically different kind of approaches in theoretical aspects we have set up for understanding the pharmacokinetic behavior of drug. So, in model independent approach only non-compartmental analysis is there. So, first we will focus on the model dependent approaches. So, type of pharmacokinetic models, as I have told you, compartment models, empirical models are there. Physiological models means realistic models, we perform it on we perform biological experiments. Distributed parameter models, realistic models again. So, first we will talk about compartment model. So, we will cover all the model dependent approaches, in which first is compartment models. So, compartment analysis is the traditional and most commonly used approach to pharmacokinetic characterization of the. It is the most common method we use in assessing pharmacokinetic profiles of a drug. These models simply interpolate the experimental data and allow an empirical formula to estimate the drug concentration with its time, right. So, we have different kind of mathematical formulation. When we observed it experimentally, then the readings we put in empirical formulas to get the desired value and the desired measurement, and on that basis we can say the drug is performing like this or not, right. Since compartments are hypothetical in nature, compartment models are based on certain assumptions. So, those certain assumptions needs to be considered throughout the experiment you have performed, right. So, hypothetical means you have set hypothesis that it will be the desired effect if we perform this biologically, whether it is matching or not, the hypothesis is matching or not, accepting or not, will give you the exact result after experimentation. So, compartment analysis is commonly used to estimate the pharmacokinetic characters of a drug, right. Pharmacokinetic characters like AUC, Cmax, Tmax, distribution, volume of distribution, clearance, all these are basic pharmacokinetics processes, right. So, the compartments are hypothetical in nature, they are not original, but we consider it when while calculating the formulas and establishing equations or models to identify certain pharmacokinetic parameters. So, we established some sort of models and consider body as a compartment. So, this is totally hypothetical. The body is represented as a series of compartments arranged either in series or parallel to each other that communicate reversibly with each other, right. So, body is represented as a series of compartment, like for example, blood is a central compartment and kidney, liver, heart, we can say different important compartment which is connected to the central compartment known as blood compartment. So, blood will be considered as your central compartment, whereas kidney, lungs, liver and heart, your vital organs will be your peripheral compartment, which is interconnected to the central compartment. Each compartment is not real physiologic or anatomic region and considered as a tissue or a group of tissue that has similar drug distribution characteristics, or we can say similar blood flow and affinity. That means, I am trying to say that when the drug is reaching to a systemic circulation, that means we are considering body as that every organ, every tissue of that particular organ is having the similar affinity or similar blood flow vascularity of vascularity towards a drug, that is drug is attaching to the protein or receptors to that particular or every we can say that every organ in similar manner. So, we consider body as a single compartment in that case. So, every organ, tissue or body fluid that can get equilibrated with the drug is considered as a separate compartment. For example, the central compartment here is your blood compartment and associated peripheral compartments are separated compartments. The rate of drug movement between compartments, that means entry and exit, from the blood compartment to the peripheral compartments, entry and exit, is described by first order kinetics. First order kinetics, how the drug is entering to the system and how it is getting eliminated or moving towards the different compartments. The study can be analyzed by identifying first order kinetics. So, rate constants are used to represent to entry and exit from the compartment. So, basically for identifying these entry and exit of a drug from one compartment to another, we use rate constants, at which rate drug is entering or exiting from the systems or compartment. The compartment models are divided into two categories on the basis of identification or the alignment of compartment. These are further divided into two: mammillary models and catenary models. So, the overall, if we talk about, the body is represented as a series of compartments arranged either in series or parallel to each other, either in series like this, like this, or parallel. Parallel means multiple in parallel. Like each compartment is not real, physiological or anatomical region, but fictitious or virtual one, considered as a tissue or a group of tissue that have similar drug distribution. That means, in that case we consider every compartment having similar type of affinity towards drug and similar blood flow. Within each compartment, the drug is considered to be rapidly and uniformly distributed. For based on that similar affinity and blood flow rule, we consider that when drug is entering to the system, it will be distributing uniformly among all the compartments. The rate of drug movement between compartments described by first order kinetics. So, you see the compartments are divided into highly perfused area or compartment, poorly perfused tissue, fat group, negligible perfused tissue group. So, classification of human body into compartments. So, the human body is basically divided into four groups, like which the compartments or organs which are highly perfused and have good flow, vascularity, and whereas some areas and organs are poorly perfused tissue, fat group, negligible perfused tissue group. So, we can say that the binding affinity of drug will be high in highly perfused area and lowest in poorly perfused or negligible perfused area. So, model dependent approaches. So, basically as I have told you that it is divided into two categories. First is compartment model. Compartment model is mammillary model, catenary model, and second is physiological models. So, physiological models. So, these are references. Once again I would like to give the highlights of this presentation, like how it is considering the body as a compartment. So, you see here set drug movement within the body. So, we have discussed that drug is moving throughout the body, entering the body and distributing in the particular pattern and then eliminating. So, for identifying these pharmacokinetic parameters, we generally set up some certain models. So, that particular models are divided into two types, like model dependent and model independent. So, model dependent approaches are those approaches in which we consider body as different compartments, understand, and whereas in model independent approaches we consider the body has no different compartments and it is non-compartmental analysis basically. So, in model dependent approach we have different type of approaches like compartment modeling, physiological modeling, distributed parameter modeling. So, These are model dependent approaches. So, in compartment modeling, subdivisions are there: mammillary, catenary, physiological model, perfusion limited model, diffusion limited model, distributed parameter model, whereas the compartment modelings are totally mathematical based models, whereas physiological and distributed parameter models are basically biological models or experimental models we use. So, here the compartment analysis is the traditional and most common method in which we consider body as a compartment having different compartments, and based on the compartments we assumes that all the compartments of the body have similar affinity towards the drug binding as well as high viscolarity or high blood flow. So, you need to understand all these points that we are, in order to identify different pharmacokinetic parameters, we need to set up one type of set of conditions to analyze these parameters. So, in that case we use models, compartment models, arithmetic models. So, these are hypothetical in nature, not real. So, for that particular compartment analysis, it is not real, it is just hypothetical. The rate of drug movement between compartment is also described by first order kinetics, means in compartment modeling we generally identify the drug kinetics through first order identification, rate constant to represent entry and exit from one compartment to another compartment. How can I explain this? You just see that if we are talking about entering and exiting of drug from the compartments, that means we are taking orally a medicine and then it is entering to the absorption phase and slowly it is going to different phase and getting eliminated from the system. That means, we consider body as a compartment, and from different one compartment, stomach, then blood, and then different compartments and eliminate it, right. So, this is how we can correlate that we are considering this concept with the compartmental things. So, basically the mammillary and catenary models are comes under your compartment models. The body basically each compartment is not real physiological or anatomical, but fictitious. Yes, we have made a base that if we consider this organ or this type of parameter to identify pharmacokinetics of a particular drug, we need to set up certain models. So, based on the blood vascularity and blood flow, basically highly perfused organs are there and present in the body, whereas the blood flow is very good, binding affinity of the drug to the receptors is very high due to high blood flow. So, poorly perfused tissues. So, it is this is the base to identify the drug performance, how it is getting absorbed through different kind of administration. For example, if drug is administering through topical route, that means reaching systemic circulation, that means in terms of bioavailability if we talk about, is less than the oral administration. Orally the drug bioavailability will be high, through the physiological variation and we can say that the different kind of anatomy, due to different kind of anatomy, different route of administration has different sort of bioavailability. So, this is basically compartment divided based on the blood vascularity and the body tissues type. So, model dependent approaches, we will be continuing this compartment model to the next session also. I hope you understand the type of models which we use for identifying pharmacokinetic parameters. Thank you, take care, and thank you for patience.